On the base of this, this is what we call the penton base. And this is an area that interacts with co-receptors, which you'll learn about in a second. So let's discuss the antivirus life cycle. And I think before I go into this, I just want you to note that understanding basic biology, not just viruses, it could be anything, it means something. You could take that information and you can apply it. So the virus I'm going to tell you about right now is literally the same vaccine vector that's being used by Johnson & Johnson and creating a coronavirus vaccine. So from researchers over many, many years trying to learn as much as they can about the virus, well, we've realized that we could use this specific one as a vaccine vector. So all that basic biology research that gets put into these systems over many periods of years can really pay dividends in the end. And this might be a vaccine that hopefully we'll see on the market in 2021 at some point. So as I mentioned, what do viruses want to do? Here's your virus. It's in the extracellular space up here, and it wants to come into the cell and release its DNA into the nucleus to create more progeny virus. So the question is, how does it do that? First, it's going to come along and it binds to CAR. CAR stands for Coxsackie and Adenovirus receptor. Remember that fiber knob that I told you about right over here? So that's binding here to the receptor. And I should mention that this is just your standard, cell lipid membrane. We have many different types of receptors, and these receptors aren't just there for viruses. They're there for all types of normal biological and physiological actions that have to happen in your cells. Viruses are smart, they're savvy, they've evolved ways to take advantage of host cells, and this is one way. So they use these receptors on the surface of the cells to enter into the cell. So first it interacts with the primary receptor. Essentially the virus is floating along. Here's your primary receptor hanging off the cell, and then bam, it attaches to it. How does it attach? It just attaches based on the amino acids in the epitopes that form weak interactions. So following primary receptor attachment, it interacts with a co-receptor, and this one is called alpha V beta 5 integrin. And again, these integrin molecules, for example, they have a multitude of different roles in the cell. Their role isn't to be there for viruses, but viruses, like I said, they take advantage of them. So following receptor binding, viruses, they have to go into the cell. And they use a process called Clathrin-Mediated Endocytosis, where they go into these clathrin-coated vesicles and then to an early endosome and a late endosome. So for those of you who have taken cell biology before, you may know a bit about endosomes and the trafficking pathways. And one characteristic of endosomes, particularly a late endosome, is that it's very acidic. So the pH is quite low. And guess what? The virus uses that to its advantage. And what it does is, when it finds itself in a late endosome, the virus says, okay, I must be in a very acidic environment right now because it's triggering conformational events on the capsid itself. And what it needs to do is it wants to open up, and it has a protein in there, which is going to be represented by a lightning bolt. And that lightning bolt is going to integrate into the lipid membrane, which is the endosome, and it's going to allow for its release. So it looks like this. So first, these fiber regions are going to be displaced, and the receptor is going to be recycled back to the cell surface. So within the virus itself now, there have been conformational changes, again, triggered by the pH environment. And there's that protein that comes across, it allows for escape, and now the virus can traffic and make its way to the nucleus. You have learned about microtubules before, and these long, hollow cylinders that allow for intracellular transport of organelles and vesicles. They use dynein and kinesin to move them around. So viruses, they utilize these organelles too. So it makes its way to the nucleus, and again, the ultimate goal is to release its DNA, and it can create progeny virus. So that's the life cycle in a nutshell. So that's one part of the story. The other part is alpha defensins. And I don't expect that a lot of you are really going to understand what alpha defensins are, or if you probably haven't heard of them, unless you've taken an immunology class before. So they are what we consider effectors of innate immune response. As Penny mentioned, they have antiviral, antifungal, and antibacterial properties. That means that their role is to defend the host cell against these foreign pathogens, and viruses fall into that category of being a foreign pathogen. So to date, so far, there have been 6 alpha defensins that have been isolated. Some are called HMP1 through 4. These are primarily secreted from neutrophil cells, which is an immune system cell. And there's also HD5 and 6. We're going to specifically focus on HD5 for today's presentation, which is probably one of the most well-studied alpha defensins. They are roughly 2 to 6 kilodaltons in size. So you may wonder, okay, what the heck does that mean? What's 2 to 6 kilodaltons? That equates to about 30 amino acids. And on the right-hand side, this is a crystal structure of alpha defensin 5. And what you notice here is I've highlighted these, I think it's about 6 residues or so far. There's 2 of them over here. So these are all arginine residues. And hopefully right off the bat, what's jumping off the page is you're thinking, oh, arginine residues, they are positively charged. So we refer to these peptides, okay, so this is an alpha defensin peptide. We refer to it as a positively charged molecule. And classically, they disrupt bacterial and envelope virus membranes, right? So we've already spoken about envelopes. They're made up of lipids. Bacteria, they're made up of lipids as well. So they have ways to integrate into these membranes and then open them up. And for like bacteria, for example, essentially lyses the bacteria. And there's an efflux of the material that comes out of it. And that's one way of, it's a killing mechanism. My last bullet point is they've been shown to enhance adaptive immune responses to antigens. So what that means is if you've ever heard of an adjuvant before, adjuvants, they can enhance immunity. So at this point, so I started not this specific work, but I was aware of these molecules during my PhD work. And the lab that I joined when I was at Harvard, they had freezers filled with all different types of adenovirus serotypes. So I was in heaven. I knew the literature, I understood the literature, and my thought process was, well, maybe I could use these defensins to try to enhance the vaccine properties of some of these adenoviruses that we're using in this vaccinology lab. So that was really the premise, and you'll learn more about that premise as we move on. So despite all that, and interestingly, so to date, there's actually very little information known about how alpha defensins interact with non-envelope viruses, which would be adenoviruses, adeno-associated viruses, and others too. So we're narrowly focusing on adenoviruses. I came across this manuscript many years ago titled Insight into the Mechanisms of Adenovirus Capsid Disassembly from Studies of Defense and Neutralization. The take-home point here is that certain adenoviruses are inhibited by defensins, while others are actually enhanced. And the ones that are enhanced, namely species D and F adenoviruses, right? So basically what all this means is we have lots of different types of adenoviruses, right? Let's just say we have adenovirus serotype 1 through 100, and they all differ based on sequence. We can put them into categories based on their sequences, and some fall into category species D and species F. And what researchers have shown is that some of the adenoviruses that fall within those categories, their infection is actually enhanced in the presence of defensins. So remember I mentioned defensins are antiviral, antifungal, antibacterial. Well, we have some instances in the literature that suggest that defensins can actually enhance adenoviral infection. So that means at some point, evolutionary, alpha defensins or adenoviruses, they decided to hijack alpha defensins and use them to their own advantage, which I think is personally really cool. So based on that information, my thought process I mentioned was we could use these defensins to aid in vaccine delivery. But we could utilize them as a potential therapeutic or an adjuvant to enhance both the viral transgene expression and then also the adaptive immune response to the expressed antigen. So that is the goal here. And again, I could just put it in a framework of what you're probably seeing in the news right now with coronavirus vaccines. So some of the coronavirus vaccines, we won't talk about Pfizer. Pfizer's using just an mRNA-based vaccine, which actually looks really interesting. Hopefully that works out. So if we use one of these vaccine vectors, the vaccine vector is essentially the delivery vehicle, and that's going to transport the antigen, which is what we call the transgene, into a patient. And that transgene essentially could be the spike gene of coronavirus, because we want to develop an antibody against the spike gene of coronavirus. So all you really have to do is you just remove the viral DNA, put in the spike gene, the transgene cassette, into the virus, and then inject that into a patient. And then if we could potentially use alpha defensins too, we might be able to enhance the immunity against that antigen. And that is vaccine delivery in a nutshell. So the advent of this work started with in vitro assays, which is what most assays would start with. It's a good way to screen for your hypothesis, and you want to identify if it actually comes to fruition or not. So we used an in vitro assay approach. And I narrowly focused on 4 different adenovirus serotypes called Ad5, Ad26, Ad35, and Ad48. All 4 of these are rather popular in the lab where I was working before, and then also in the adenovirus vaccine delivery field. So what we did here was, you know what the adenovirus looks like. We removed the adenoviral DNA, and we packaged it with something called EGFP. EGFP stands for Enhanced Green Fluorescent Protein, and probably some of you know that already. If you've ever worked with cells and tissue culture and expressed a protein, and all of a sudden you looked at it under a specific blue light and you saw cells were glowing green, these EGFP transgene cassettes they may be the reason why that was the case. So that's what we use, and that's our output. So we're essentially going to look to see how much EGFP expression occurs from each one of these viruses. And of course, we do that in the context of alpha defensins. We do it with defensin present, and then also with defensin absent, because we need some sort of control or a comparator. And you'll see why that's important in a second. So we complex the defensins and the viruses together. We incubate them for 1 hour on ice. And then we infect what we call A549 cells. These cells are very permissive to adenovirus infection, which all that really means is that adenoviruses can infect this cell type. So researchers all over the world who study adenoviruses, they will use A549 cells along with other cell types too. So once the infection is complete, we allow that to occur for about 24 hours. So 24 hours post-infection, we're going to assess, which means monitor, for our EGFP expression, which is the green fluorescent protein. And this is all of our quantifiable data shown right here. So this is obviously a busy diagram, and there's a lot going on. So let's break this down. We have our 4 different adenoviruses at 5, 26, 35, and 48 that are color-coded down here. On the y-axis, this is what we call percent of control infection. This is how much EGFP is being expressed compared to the wild-type virus. So what wild-type virus would mean, if you look on the x-axis, we have these different numbers. And each one of these numbers, it corresponds to how much alpha defensin 5 we're using in each experimental condition. So let's look at Ad5 first. We have some controls over here, but we don't really have to focus on that right now. So we have the number 0. This means this is wild-type adenovirus 5 at a very specific concentration of virus. And what we've done here is we infected our tissue culture cells, and we looked for EGFP output, and we normalized that to 100%. And then we asked the question, which adenoviruses in each one of these experimental conditions, but the difference is, so it's not the concentration of adenovirus, the difference is the concentration of alpha defensin 5. What you see is we go from wild type to 200 nanomolar to 2 micromolar, 8 micromolar, 16 micromolar, 33, and 50 micromolar alpha defensin in each experimental condition. And the trend shows that alpha defensin is actually inhibiting EGFP expression from adenovirus 5, again, right, adenovirus 5, with increasing concentrations. So that's the phenotype that we see with Ad5. We also see the same phenotype with Ad35 with higher concentrations. There's less and less EGFP being expressed, which again is a measure on the y-axis. And with Ad26 and Ad48, these are in contrast to that. And when we compare the two, Ad26 goes up and Ad48 goes up even more than that. which is really striking and really cool. So for me, this is when that light bulb went off and I became very excited with this research because I thought I may be onto something. You have to keep in mind, so when I started working in this lab, which was around 2014, this Adenovirus 26 vaccine vector was really hot vector in the vaccine field, and it still is, right? So Johnson & Johnson's using it now. It's been used for Ebola vaccines. It's been used for Zika virus vaccines. So if we can actually improve the efficiency of this, then that would just simply be awesome, just from a passionate scientific standpoint. So taking this information, which showed in vitro my theories and my hypotheses that came to fruition, as you would guess and surmise, an in vitro experiment is not enough, right? So we use these vaccines in humans. So you mean to tell me that if I just show this in vitro in cells, that's good enough to say this vaccine is going to work in humans? No, so it doesn't work like that. So what you have to do is we're going to test this, the same sort of theories and experimental conditions in animal models. So that was my next step. And that's why I went into what we call, this is an in vivo mouse model. It's called Ibis Imaging. And the way this works is, so here's a mouse, and we formulate these syringes with our adenovirus, with and without defensin, and we inject them into mice. So the procedure works like this. We perform IM, which stands for intramuscular immunizations. Just think about when you've received influenza vaccines in the past, right? You get the vaccine up here and that goes into your muscle. That's a standard location for vaccination. And we're narrowly focusing on two different adenovirus serotypes, Ad5 and 26. So remember, Ad5 with defensin is inhibited, Ad26 is enhanced. We're also using luciferase as our measuring output. And luciferase, you could think of, if you've ever seen luciferase, the fireflies, right, in the summertime, it's essentially the same enzyme that's being expressed. So it's a measure of light, and that's how we could detect it. So with that said, we also have something called luciferin, and luciferin is what serves as the substrate for luciferase. So once they bind to one another, you see a lot of glowing that occurs, and you'll see that on the next slide. And our timeline for this, we immunize the animals at day 0 or time 0, and we would assess with luciferase treatment and the luciferin treatment and Ibis imaging at different time points post-immunization. So 6 hours, 1 day, 3 day, and 7 days post-immunization. And what we're really looking for, we want to see if the light expression is going up or down in these adenovirus 5 and 26 vaccine vectors when defensin is present or not present. This is what the mice in the setup look like. And this is a day 3 representative time point. So on the left-hand side, this is adenovirus 5. This would be Ad5 alone. And we injected the animals in the hind leg, shown over here. And these little dots, and it's cyan, some really bright colors. This means that luciferase is being expressed. So you can see it, and I'll show you in a second how we quantify it. So in most of these animals for Ad5, we see expression, which is great. We also see it with Ad5 and what we call the mutant HD5, which is a negative control. So it really shouldn't have any impact on viral expression. And that's what we see. And then we ask the question, as we did before at the in vitro experiment, we have Ad5, then we're going to increase the concentration of defensin up to 25 micromolar, 50 micromolar, 100 micromolar. And then we do a control, which is PBS. This is essentially, it's almost like a placebo group, and you guys have heard about placebo groups. So these are all the different types of groups that you have to put together for an experimental setup like this, right? It's not cheap to run mouse experiments, so you want to make sure that you go into it with the know-how and the information that you need to conduct a really good experiment where you don't have to go back and keep repeating it over and over again because you may have forgotten a control group. Ad26, the same experimental setup, and the data is quantified on the right-hand side. This is adenovirus 5 here. Total flux, this corresponds to luciferase expression. So the higher the value or the points over here, that means the more luciferase is being expressed. And on the x-axis, these are days post-immunization. And just a little caveat, I think it's really important when you give scientific presentations to point out the y-axis, x-axis, it's very easy for people to lose focus, not follow if that information is not pointed out. So you want to try to make it as like blatantly clear as you can for the individual, just FYI. All right, so for AD5, we have AD5 up here in this color, and that's shown designated by this line right here, and that is our normal level of expression. And when the mutant peptide was in that same mixture, in that same experiment, we see that there's really no difference between the two. So NS stands for no significance. However, when we increase the concentrations of normal wild type alpha defensin 5 going from 25 to 50 to 100 micromolar, we see these concentrations, we see the luciferase expression almost essentially completely abolished, and it's really baseline, which is our PBS control shown down here. So it's essentially the same. So this means that it corroborates our original data, the in vitro data that showed defensins can inhibit adenovirus 5 expression. And for AD26, in contrast to what we just showed for AD5 phenotype, it's the complete opposite. And that's really good. We want to show that we can enhance AD26 expression, specifically luciferase. And the highest concentration of AD26 with 100 micromolar of HD5 is shown up here. And that is statistically significant compared to our wild type, which is shown down here. So this data was very revealing. It suggested that the data translated to what we saw with the in vitro work that we had conducted. So from here, we thought we were onto something. Then we said, okay, well, let's just actually test the true vaccine properties of this vector. And as you may have heard in the news recently, when we use vaccine vectors, what we're trying to do is either stimulate IgG or antibody responses or T cell responses. So I'm going to take you into the depths of both of these right now. So first, we want to see if we could stimulate antibody expression in this mouse model system. So here we used what we call black 6 mice. It's a slightly different system than the other mouse, which was white, which is called BALB-C. And we immunized the mice with either AD5 or AD26 with and without defensin. In this case, they're expressing something called SIV MAC239ONV. And what that really means is this is the envelope gene that's used in SIV, which is simian immunodeficiency virus. And 239 just corresponds to a specific monkey number that was identified many years ago. So what we're trying to do is express this envelope protein and then develop antibodies against it in the mouse. So after we immunize the animal at day 0, we assess for antibody expression at days 14 and 28. And we do this by bleeding the animals. And we isolate their serum, and then we test their serum, which should have circulating antibodies using an experiment that we call an ELISA. And again, we have, if you've heard about the Pfizer clinical trial, there's 40,000 plus people that were part of this, and are using these mRNA vaccines to introduce the actual vaccine itself, right, the antigen. And what they do is they follow these individuals in these clinical trials out for extended periods of time, and they'll come in for blood draws, and they will be assessed. Their blood's going to be assessed for antibody responses and also CD8 T cells, sometimes CD4 T cell responses. So what we're doing here is a typical setup that you would use in the lab, more in the introductory phases of your specific experiment when you want to test a theory or a hypothesis. All right, so this is the data, and the data corroborated everything that we've seen so far. Up here for AD5 on the y-axis, we're looking at antibody responses. So this is antibody responses here. On the x-axis, we broke it into 2 groups. Remember I said we assessed these animals, we bled them at 2 different time points, day 14, and then also day 28. This is the wild type virus shown here, which means this is just AD5 alone without any defensin, which is shown in black. And then we added increasing concentrations of defensin, something that we've been doing. It's a general theme in all the experiments. So we went up to 25, 50, and 100 micromolar. And with each increase, each increment of defensin, there was more and more inhibition, which again is all statistically significant. Day 28 followed the exact same trends, albeit the levels were a little bit higher, and that's what you would expect with antibody expression. Now for AD26, we were looking for the opposing phenotype. Here we have our day 14, our day 28, and these values, each one of these values responds to a different concentration of adenovirus. We go from 10 to the 6th to 10 to the 7th to 10 to the 8th VP per ml of adenovirus. That's shown over here. When we look at day 14, the 10 to the 6, we see there's absolutely no difference between just adenovirus 26 alone versus adenovirus 26 when defensin's present. Now, there is one animal that did have a relatively high level. So who knows if that data point is real or not? It's hard to say. So then we look at the next group. And the next group is this is our baseline value of adenovirus 26 expression at the 10 to the 7 concentration. When defensin's present, it goes up and it's statistically significant. So what this tells me, there's an early time point here. And if these antibodies, so right there against SIV-ONV, let's just say it's against the spike protein in coronavirus. So if they're against the spike protein in coronavirus, they may be able to show efficacy at an early time point. So if I were to use this vaccine setup with defensin as an adjuvant, you guys may be covered at your day 14 time point compared to not having defensin present. And these are the thought processes that you need to kind of keep in your mind when you're thinking about these experiments. The 10 to the 8th concentration, there was no statistical significance, really nothing different between the two. When we look at day 28, and again, the upward increases of concentrations, we saw there were a few animals that did have elevated levels of IgG responses, 10 to the 7th and 10 to the 8th, no real difference. So the take-home point for this data is we were able to inhibit IgG expression of this antibody for adenovirus 5 with increasing concentrations of defensin. And for Ad26, we're able to augment, which means enhance, the antibody responses when defensin was present compared to just wild type virus that doesn't have defensin. So this was really, really, made me very, very happy. So from here, I went into one more aspect, and we actually have a couple other aspects in the manuscript, but not really going to discuss them today. So next, we went into what we call Listeria challenge model. Think about this. You've probably heard of Listeria before, and you may know that it's a bacteria. Every once in a while in the news, there's always a news cycle where vegetables, produce, spinach, lettuce, it gets contaminated with Listeria, right? So that is a bacteria that could cause it could do some nasty things. So we actually have a model, an animal model system that we use in the lab that we could use Listeria as a challenge system. So I'll discuss that right now. We used the black 6 animals again, and we immunized these animals with Ad5 or Ad26, expressing what we call ovis and fecal. And all this means is, so you have Listeria, and the vaccine for Listeria is this ova synfecal. So if you inject the animals with these vectors that are carrying the ovis and fecal DNA, and it starts making protein for ovis and fecal, if that's circulating in the body when the animal gets infected with Listeria, then there's not going to be any infection, right? This is protection. This is efficacy. So that's what we're trying to do. We want to see if using Ad5 and Ad26, these ovis and fecal vectors with and without defensin could protect against Listeria infection. And this system, we specifically use CD8 T cells. Now this is Listeria can be protected against by CD8 T cells, which is why we assessed and monitored for these specific T cells. So our timing on this was we immunized the animals at day 0. We do something called tetramer staining, which we don't have to go into, but all we're really doing is we're assessing the animals to see how much CD8 T cells are present. And you'll see that on the next slide. And at day 12, at this point, the animals have been vaccinated. They should have CD8 T cells that are primed against the ovis and fecal, which means they're going to have protection against Listeria. So then we challenge them with Listeria at day 12. And then within a couple of days of that, we look to assess how much Listeria was actually growing in those animals, or if we're able to essentially abolish it and prevent infection from occurring. And this is what the data looks like. On the top panel, we're examining CD8 T cells. On the bottom panel, this is looking at colony-forming units of the Listeria pathogen itself. So I'll break it down for you right now. We have Ad5 here, and this was done at a concentration 10 to the 8th. We have it without defensin, which is shown by the minus sign, and then with defensin, which is shown by the plus sign. So our CD8 T cells, which is this is our baseline, right? This is our wild type adenovirus. We're able to decrease these values when defensin was present. So that means you should have less protection against Listeria when defensin is present. So now let's assess and examine Listeria infection. So our normal wild type shown up here, we had this amount of Listeria that was present, which is between 10 to the 0 and 10 to the 1. So it's a relatively low amount. When we compare it to Adenovirus 5 with defensin present, the Listeria concentration went up a lot. And that makes sense because we were abolishing a lot of the CD8 T cell responses that should have protection. So this data is falling in line with our general theme so far. Now, additionally, for Ad26, we're expecting the opposite phenotype. This is our baseline for Ad26 CD8 T cells. And with defensin present, we were able to augment it quite a bit, and that was statistically significant. And when we compare that to Listeria with Ad26 and defensin with these very high CD8 T cell levels, we essentially didn't have any Listeria expression. This was just baseline background. But when defensin wasn't present, there was a very high count of Listeria. And then we took it up for Ad26 at other concentrations. And we do have some partial significance here and there, but the take home point again is we're able to show protection by priming CD8 T cells and enhance and augment the vaccine delivery properties of Ad26. So in summary, what we've done, we've expanded the previous comparative findings on species D and F adenovirus vectors, and that's based on that manuscript, the title that I read to you earlier. And we did that by providing additional data showing HD5 augmented phenotypes for Ad26 and Ad48, which hasn't been done before. And our data also indicates that Ad26 antigen-specific CD8 T cells, this is our ovus and fecal, and our humoral IgG responses, and that, again, that's against the SIV MAC239-OV, we could enhance them by HD5. So with all manuscripts, when you submit a manuscript for review and for publication, they want to know about mechanisms of inhibition. So sometimes identifying a mechanism of inhibition or enhancement is one of the difficult things you could do. In this case, there was some information already in the literature. So the mechanism of inhibition looks like this. If you remember the life cycle slide that I showed, and I showed those fiber regions that extend off the virus, this is what that fiber region actually looks like. I also mentioned the base of the fiber region has that penton base, which we call, the penton base is essentially what we call an RGD motif. I'm just showing that over here. So this is the base of the fiber. This is the fiber up here. Remember I told you the end of the fiber, which is called the fiber knob, it interacts with the primary receptors. So that would be in this location. And what alpha defensin is doing, HD5, which is shown in red, it binds to the base in this little pocket. And during the life cycle stage, when the virus is in the endosome and it's triggered by the acidic environment and it displaces these fiber regions, which are necessary so that lightning bolt can come out and open up the endosome and the virus can escape and move to the nucleus via microtubules. Well, these defensins, they inhibit that process. They prevent that from happening. So the virus is rendered in the endosomal state. And this epitope right here, and this is the significance of really understanding the central dogma of science and biology. So this epitope here that's shown in red, that corresponds to this region here. These are the amino acids that make that up. And I grouped these adenovirus serotypes by ones that are inhibited by alpha defensin 5 and those that are augmented by alpha defensin 5. And what we see is there's some characteristics that distinguish the two groups. So number one, the HD5 inhibited serotype, so Ad5, shown over here, that falls into that category. They have these negatively charged amino acids, right? Aspartic acid, glutamic acid. You've learned about these before. So they're negatively charged. The thought process is defensins, they can bind to this pocket because alpha defensins are positively charged. I mentioned that they have a series of 6 arginine residues. We consider them cations. So the cation properties of it binds to this fiber region, to the base of the fiber region. It prevents displacement of this. Thus, the virus cannot encode and go through the rest of its normal physiological processes to deliver the DNA into the nucleus. So it stays in the endosome state. Now, when we compare this to the HD5 augmented serotypes, they are devoid of those negatively charged amino acids. So you don't see those over here. And the thought process is that they don't have them there. So this is not a standard epitope where they may be binding on Ad26, for example. But defensins are obviously doing something. So there are some theories out there, although we don't fully know. So one of the theories, and what's been shown before, and it's the first bullet point, think about when you learn about your lipid membranes, and you have those phospholipid heads and the fatty acid tails, right? We all know that lipid membranes, in general, they're very hydrophobic. But the phosphate groups on the surface, the phospholipid head, that's negatively charged, right? So there's some interaction there that can take place with the aqueous environment. In this case, what we're thinking is happening, the defensins are binding to different pockets on the outside surface of the virus, and it's neutralizing those negative charges. So that means instead of this very negatively charged virus coming in close contact to the negatively charged phosphate groups, and there's a repelling event, well, now we can neutralize some of that, and there's an uptake or an increase, an enhancement in the amount of virus that gets into the cell. So that's one of the theories. I'm not really going to discuss the second theory. And one of the other questions is, are there alternative trafficking pathways? So there's a lot of work that can be done with this. So maybe the virus isn't going through those Clathrin-coated pits to the endosome. Maybe it's found a different way when defensin is present. So there's a lot, the more research you do, the more questions you ask, which is the beauty of it. And we have a final model, and to answer our specific question that we mentioned earlier. So when we compare Ad26 and Ad5, and we have our alpha defensins that are located here. So that's shown in these little red molecules surrounding the virus. Here's Ad26 and Ad5. These are in the endosomes. Well, what we do know is Ad26 certainly doesn't have any issue escaping and leaving the endosome. So it has that ability. So it moves to the microtubule, into the nucleus, and then it releases its DNA. On the other hand, Ad5, as we mentioned, because it can't displace those fiber regions, it gets stuck in the endosome. So the original question that I posed in the beginning, were alpha defensins friends or foes in adenovirus infection? I think it's safe to say that they're a friend to Ad26, but also a foe to Ad5, right? So Ad26 has the phenotype that is enhanced and expressed. Ad5 is inhibited. So with that said, I want to thank my former lab, Dan Baruch, who's a fantastic scientist. I had a lot of fun in the lab. I was able to do a lot of really cool and interesting research. It was a really wonderful experience. And, but more importantly, I want to thank Nandini, Penny, Tracy, and Anna for the invitation to be here today. This really means a lot to me. I'm so happy to be part of this in some way and offer advice and with anything I can. So thank you everyone for your attention. I really appreciate you coming today. I know a lot of you are busy. Some of you have a genetics exam in my class tomorrow. So thank you for showing up. Really appreciate it. And I'd be more than happy to answer any questions you may have.
Yeah, so like Dr. Tartaglia said, we're going to be going into a short Q&A session. So if you have any questions for him, you can ask them now. So just go ahead.
Hi, go. Well, I want to know, I haven't taken a biology class yet. I'm in my first semester here, but what is the point of an endosome? Is it to kill the virus with its acidic environment?
No. So you have endosomes that are naturally in the cell, and you have different things that happen in the endosome. So you can look at endosomes in different ways. So in some cases, you're actually putting it inside the cell that's going to allow for misfolded proteins, other types of pathogens to be broken apart, right? So that happens. In the acidic environment, as I mentioned, you're going to learn about the folding of proteins, and sometimes proteins get misfolded. In ways that we could really misfold them is we put those in those environments. But endosomes, they are natural processes. They're natural compartments in the cell that could transport different vesicles and things from location to location. So we have those in the cell. So they weren't naturally made against viruses, but viruses have been around for so long that they've evolved a way to take advantage of those two.
I have a question. So why did you choose to go into this field of research?
The question was, why did I choose to do this type of research?
Yeah.
Yeah, that's a good question. So part of my master's degree, I was working on ice worms, right? And some of you guys know that worms that live in glacial ice and snow, and they survive at 0 degrees Celsius. They're called extremophilic organisms, specifically psychophilic organisms. And we want to identify what physiological adaptations permitted survival for them. That was like really basic biology research, which when I say basic, it doesn't mean it's like easy research. It just means it's like very basic research. So I wanted to do more translational type stuff. So through my PhD, I moved into studying viruses and using them for gene therapy vectors. And I really enjoyed it, and I wanted to take it even further. So during my postdoc, I joined this high-powered lab, right, who's in collaboration right now with Johnson & Johnson. We're the first lab in the world to develop an effective Zika virus vaccine that was shown in animal models to have protection. So for me, I've always had an interest in viruses and using viruses in different ways and understanding the basic biology properties of them, but also ways to enhance them, make them more efficient for different types of models, right? And if we could use those in humans and create a better vaccine, and my part, like I feel like I'm contributing to humanity, which that makes me feel good. But I love viruses, I love studying them, and so that was, that's really where this interest came from.
Anyone else have any other questions?
Hey, Dr. Larry. Earlier, you talked about how you inoculated the virus over ice. Is there a reason why the virus would prefer to be inoculated over ice rather than physiological temperature?
Yeah, good question. So I want you to think about, if you've heard recently with the Pfizer vaccine, right? It's an mRNA-based vaccine. So it's mRNA. And a lot of you guys have learned that mRNA is unstable. And the reason for that is because the 2' hydroxyl group, which could interact with other things. It's not DNA, which has the secondary structure, or just the structure in general that has the double helix that keeps stability, right? So mRNA is it's like this naked nucleic acid, which has to be kept at very, very cold temperatures for stability. It's the same thing with viruses, and not all viruses, but adenovirus in particular. What can happen sometimes, if it's out at room temperatures for extended period of times before you actually start your experiment, then you're going to run into an issue. Those fiber regions, sometimes they just fall off, and there's no rhyme or reason why it happens, but it can happen. The fiber is needed for cell surface receptor attachment and so forth. So the best thing that you can do is really, it depends on the experiment, what virus you're working on and what those conditions call for. But for me, at least in my hands, it worked best to actually perform that experiment. You incubate it on ice for an extended period of time. It doesn't actually take long for those interactions to take place with the defensin binding to the virus. But you give it enough time where it stays like that. And the idea, if we could keep those fiber regions on the virus and keep it stable, then when we actually inject it onto the cells and we allow for infection to occur, then at that point, sort of what you just mentioned, we put it in tissue culture incubators at 37 degrees Celsius, the same temperature as our bodies, has certain levels of carbon dioxide and so forth. So it's a happy environment for the cell, and the viruses, they could do their work and do their infection then, but you want to prevent any type of mishap that can happen. So in some ways that's very specific to adenovirus, and not every single experimental condition would call for that.
All right, so if no one else